Hello friends, myself Amit Belikar welcomes you to my channel Tips and Tricks by AMB. Today we are discussing about mechanism of action of erythromycin. After going through this video, the learner will be able to describe mechanism of action of erythromycin. Erythromycin is a macrolide antibiotic consisting of a large lactone ring to which a sugar moiety is attached. It is a fermentation product of fungus Streptomyces erythreus. To understand the mechanism of erythromycin, we should know how the protein synthesis can take place in bacteria first. For protein synthesis in bacteria, double-stranded DNA strands unwinds first. This process is called as unwinding. Then enzyme named RNA Polymerase reads the portion of one of the DNA strand which is having codes for the synthesis of proteins and duplicates the portion of strands which is called as mRNA. This process is called as transcription. mRNA is having codons on it to arrange the amino acids in sequence to synthesize proteins. Ribosome is responsible for reading these codes and attach amino acids in sequence to synthesize proteins. Ribosomes are having two subunits with 30S subunit and 50S subunit. 30S subunit is responsible for reading codes on mRNA and adding specific amino acids in sequence. Thus it is responsible for initiation of the process or initial tRNA and ribosome complex formation. 50S subunit is having three sites. First is A site, which is called as acceptor site, which accepts the specific tRNA amino acid complex as per codon read by 30S subunit. Second site is the P site, where actual transfer of amino acid can take place from one tRNA to other tRNA. Transfer from amino acid from tRNA takes place and transferred amino acid joins together by peptide linkage. Third site is called as E site, which is also called as the exit site, from which tRNA leaves the ribosome. Thus 50S subunit is responsible for accepting tRNA amino acid complex, transferring amino acid and linking them with the help of peptide bonds. Then ribosome binds with one of the end of mRNA, specifically 30S subunit binds to mRNA strand and reads the codon present on it and attracts the tRNA having same codon which carries specific amino acid. This is called as tRNA aminoacyl complex. tRNA binds to acceptor site of 50S subunit and ribosome moves to the next codon. Now tRNA at the acceptor site moves to P site of 50S subunit and then leaves ribosome through E site of 50S subunit. While leaving behind, it removes or leaves amino acid which binds to other amino acid with peptide linkage. Thus a long chain of amino acid is formed, which is the primary structure of protein. Now we have to discuss about mechanism of action of erythromycin. Erythromycin is having affinity for 50S subunit of bacterial ribosomes and bind with acceptor site. Thus it inhibits transfer of aminoacyl-tRNA to acceptor site and thus also inhibits bacterial protein synthesis by preventing incorporation of aminoacyl-tRNA at mRNA. It also prevents the formation of peptide bond. Thus it produces bacteriostatic action. Now it is time to summarize. In this video, we had discussed about mechanism of action of erythromycin, and we had also discussed how it produces bacteriostatic effect in bacteria. So thank you for listening me patiently. If you have any queries, you can contact me on my mail address which is given below. If you want to watch more such videos, please like and subscribe my channel Tips and Tricks by AMB. Thank you once again.